Welcome to Click for IB Biology. We're studying topic B 3.2, transport. We're looking at a transport system in plants which takes place in the phloem, which is known as translocation. There's a PDF of this presentation that's linked into the description down below the video. Download that and annotate with your own notes as we go along. But then there's a quiz of 10 questions with answers that you can use to check your understanding of this part of the syllabus. So translocation is a process that takes place in the so-called phloem tissue, and it's responsible for moving molecules, which are often referred to as photosynthate molecules. Now this includes soluble sugars such as sucrose, amino acids, and oils. How to move these molecules around the plant from the leaf when on occasion it's necessary for that movement to send them downward, perhaps into storage structures within the root, and yet the same system is capable of sending materials up to the growth point of plants, and indeed it can reverse the direction in which the flow is taking place. The phloem tissue, which is associated with this particular process, is located on the outside of the cambial tissue and towards the cortex tissue, as seen here in this region of the cambium. With a light microscope, we can look at a cross-section of the cambium, and characteristically these larger rays of cells here, these are the xylem, and our phloem is out here, and immediately we recognize that the cross-sectional area of these phloem tubes is considerably less than that of the xylem. Indeed, if we look down into the next diagram down here, still in cross-section, we can see the end of one of these phloem tubes with its sieve plate in this location here, and the diameter of that is indicated by our scale bar here. If we look at the mechanism for the movement of these organic molecules within the phloem, we are now looking at longitudinal section up and down the plant, and the phloem is defined by a series of cellular structures in which the cytoplasmic content of the cells is largely lost. There is some remaining cytoplasm, unlike the xylem, which has lost everything. And the end walls are not completely broken down as they are in the xylem. And so we have this perforated end wall structure, which is often referred to as a sieve plate, which is shown in this electron micrograph here. If we move across to the light microscope image, we can see this sieve plate in this location here, and then again it's here and indeed here. And then we also notice that in close proximity to this tubular structure known as the sieve tube, an additional structure known as companion cells. In my diagrammatic representation in the center, these companion cells have been drawn smaller so that we can get more information onto the actual image, but in fact they tend to extend the entire length of the sieve tubing, of the phloem tubing, and indeed notably tend to overlap at the junctions like so. The companion cells are metabolically active. They have a full complement of organelles and are noted for a large number of mitochondria. And so this alerts us to the fact that there is some kind of active process going on in these structures. In general terms, what is actually happening if we follow the purple arrow direction of translocation is that the upper companion cell is responsible for actively loading the organic molecules into the phloem, a process called loading, and then it's also responsible for unloading the organic material at the sink structure where those molecules will be stored. And of course, in my diagram, I only have 2 phloem cells, but the reality is that they could be as far apart as a leaf and a root structure, so much further differences. The movement of the sap through the actual phloem structure occurs by a process which is known as mass flow. The explanation for mass flow is best accounted for in a combined model which combined with Xylem, so that we can account for the pressure changes which are known to exist. So of course we've noted that in the leaf we have a source of the organic molecules. For the purposes of this discussion, let's describe that as being glucose. So glucose is produced in the leaf and we wish to transport it to other parts of the plant. The glucose will actually not be transported as glucose, as a reducing sugar. That's not particularly useful, and so it's converted into non-reducing sugar yet soluble form of sucrose. The sucrose is loaded by passing through the companion cell. The companion cell will take up the sucrose from the leaf cells and pass this into the space of the phloem tissue, and in doing so it passes through what are called plasmodesmata, cytoplasmic connections between the companion cell and the phloem. And this process is an active process which uses up ATP, which is generated in the very many mitochondria which are seen to be located within the companion cell. And so, in doing so, this creates a very low water potential within the upper region of the phloem tube. The close proximity of the xylem means that water which is on its way to the leaf is diverted and taken up because of the water potential gradient and fills the phloem tube, placing the sucrose in solution, but also increasing its volume and therefore increasing the pressure. And this is the pressure that pushes the sap forward by the process that we referred to previously as mass flow. I should have mentioned that the content of the phloem, the dissolved molecules, are generally referred to as the sap. So the sap is under pressure, generated where there's loading, and then passes down through the sieve tubing to the location in which it will then be unloaded by more companion cells for storage. This unloading process is actually not an active process. The removal of sugars from the phloem into the companion cells is actually based on diffusion gradients. However, ATP is used when transferring these sucrose molecules into the other cells that connect it to the storage tissue, such as a tuber structure in the leaf. That sucrose will then be converted into an insoluble form, such as starch, where it will not disturb the osmotic balance of the process in the root. Now, the removal of sucrose from the phloem then leaves behind the water, and then this is taken up again once more by the xylem vessels, and it forms part of the transpiration stream. And so you will notice that in order to have a full explanation for translocation, it's necessary to also be aware of the role of the xylem, and in fact the two systems are working in concert with each other in order to create the translocation process. That brings us to the end of the topic B3.2. There's the chance to subscribe to the channel. That's always inspiring if you do that. and there's the playlist so that you can access all of the transport videos in this particular sequence.